Today we're going to be doing a short lecture on just the anatomical features of the spinal cord, chapter 16, and this would be part 1. So when we look at the general divisions of the spinal cord, what we're going to see is that we have a cervical plexus usually. It's going to terminate about C7, so from C1 to C7. We have a thoracic plexus, which is going to terminate from T1 to T12. And then we're going to have a lumbar plexus, which is usually L1 to roughly about L5. A sacral plexus, which is usually S1 through S5. And then the coccygeal can be usually anywhere between 1 and 3 bones of the coccyx. So some important aspects to note is that around T12L1 we see the change of the conical. So as we start to see it, it'll begin very large and it'll be cone shaped and it'll taper as it drops down. There is a structure called the filament terminae. This filament terminae extends from basically the second sacral vertebrae and connects into the conus medullaris. So this helps anchor the spinal cord in the lower half of the lumbar and the sacral plexus. This whole region is called cauda equine. They call it the horse's tail. As you can see here, it kind of resembles fibers as it goes through the changes. So just the general characteristics when we're looking at the differences in diameter. The largest part of the spinal cord is essentially, when we're looking at the transverse diameter, we're looking at the division between the left and the right side. So we're measuring here, and usually in the cervical plexus, which is the largest, it hovers around 13 to 14 millimeters. When we're looking, it has a larger portion of white matter, so here, compared to the gray matter, so you can see the relative size difference. When we go down into the thoracic plexus, you can see as it tapers, we go from 13 to 14 millimeters, and we're going to go to 9 to 11. We also see that, again, the white matter has a higher proportion than the gray matter. And this is the first time that we start to see the horns, where we see the posterior horn of the gray matter, the anterior horn, and the lateral horn. In the first thoracic, T1, this is where we start to see the beginning of the distribution of the horns. When we go into the lower lumbar region, you'll notice is the white matter is greatly reduced versus the gray matter. It's also 11 to 13 millimeters in size. The anterior and posterior horns of the gray matter are extremely large, and we don't really see much of a lateral horn in the lower lumbar aspect. Also, what we see in the sacral plexus, we see the anterior and posterior horns again are relatively large in this picture, as the same as the lower lumbar. So when we start looking at the divisions, particularly the divisions are very similar. The meninges, the different connective tissues are very similar in the central nervous system between the brain and the spinal cord. We'll start with the outermost meninge, which is called the dura mater. This dura mater here, and from the dura mater to the outside portion of the vertebral column, what we'll see is that it has this epidural space. So the dura mater is called the tough mother. It's a very tensile strength strong. It has a lot of dense collagen fibers. This epidural space actually has a couple of interesting aspects. It has the areolar connective tissue, it has blood vessels, and some adipose tissue, as you can see here, that helps absorb shock. Usually when we see injections in this region for like a nerve block, it's only going to affect the sensory nerve closest related to this spinal nerve. The next region as we go down, it's pretty small, but when we're looking at it, is the arachnoid matter, which I'll draw a very thin line down here. And you can see between the arachnoid matter and the dura mater, there's this little open space, and I'll just draw a circle over the top. This arachnoid matter or the subarachnoid space helps for the CSF. So when we're really looking at the subarachnoid matter, we're talking about the cerebral spinal fluid. And the cerebral spinal fluid is important because it offers a little more shock absorption. So it allows for this area to provide for more shock absorption. It's a diffusing medium for dissolved gases and also waste products, nutrients, and chemical messengers. They usually use this subarachnoid space for what they call a spinal tap. And a spinal tap is where they're actually drawing out the cerebral spinal fluid out of this region here, that little small space. And when they're drawing out, they can check for different types of viruses like meningitis or something like that. And then finally, the last one that we're going to look at is the pia mater. So this is the one that's actually going to be going around the full circumference of the spinal cord. With that pia mater, what we also see is the beginnings of a denticular ligament. So when we're looking at the denticular ligaments on both sides, these are just going to help anchor the spinal cord to prevent any lateral or excessive lateral movements. So the next aspect is just a different version of this. So when we're looking at the outside, we see the dura mater first. So you can see how the dura mater actually completely goes over the top of the spinal nerve. So the spinal nerve would be here, remembering that the spinal nerve is a mixed nerve. So a mixed nerve is going to contain both sensory and motor. And that's where it gets its name as far as a mixed nerve. So we have this tough collagen outer shell to help hold the position of the spinal nerve. So sensory information that's coming in through the dorsal aspect can then eventually leave out the motor aspect of the spinal nerve in the ventral compartment. Then the next layer, we see this subdural space that's between the arachnoid mater and the dura mater. The subdural space is where we would do an epidural block. And then the next one, the subarachnoid space, is going to be between the pia mater and the subarachnoid space is where we see the cerebral spinal fluid. You can see the rootlets, which we'll talk about. These are anchored in to the pia mater also. So these rootlets are anchored into this pia mater. They're actually going to go all the way into either the white matter or the gray matter, depending on where they terminate. But these rootlets are going to be important. All sensory information is going to be coming in through the dorsal aspect, either to the gray matter, communicating to the white matter, and then all motor responses are going to be coming out of the ventral or the anterior compartment, and then this is going to have the motor. So when we're looking at the divisions of the spinal cord or the spinal nerve, we usually have sensory coming in from the back and motor coming in from the anterior compartment. We can also start to begin to see the differences of the gray matter and white matter, which we'll talk about here in a second. The other important aspect of a spinal nerve is understanding the rootlets. Just like when we were talking about synapses, these are very similar to the dendrites. The more sensory information that's coming in through the posterior root can find its termination point wherever it needs to lie in the posterior aspect. And the same thing, any information coming from the brain for motor command, it can come down, or multiple pathways for motor information that eventually go out somewhere to what we consider to be the effector, so how it's going to make its change. When we're looking at the different types of white matter, so white matter, again, just for this, is divided into funiculi. So white matter are going to contain the axons. When we're looking at the white matter, we'll have the posterior funiculi, we'll have the lateral funiculi, and we'll have the anterior funiculi. So inside each one of these funiculi, there's going to be multiple tracts. So when we're looking at the different tracts of the white matter, tracts are just bundles of axons in the central nervous system that are relatively uniform. They're myelinated, and they have the same conduction speed. So again, all these different tracks here, they're all going to relay the same information. So if it's coming in the posterior compartment, all these will be sensory, and if it's coming down from the brain, then all these different tracks will be motor. An ascending track, you can also think of it that way. The ascending means that it's going to be going up, so usually it's in the posterior compartment, and descending means that it has a motor command, and it's usually going to be descending from the brain, and again, eventually out through to its effector. So whatever is going to be changed by that action potential. When we look at the different types of horns, we're going to see that there's going to be regions. So whenever we start looking at the posterior horns, particularly here, so this posterior horn, it's going to contain the somatic and visceral sensory. When we look at the lateral horns here, these are usually only located, the lateral horns, again, just to reflect, from T1 to L2. So lateral horns are only present in T1 through T2. These are going to be predominantly the visceral. And then when we look at the anterior horn here, they're going to contain the somatic, or the motor commands. So again, sensory, this would be kind of the autonomic nervous system, and then this would be the motor. And the next slide kind of shows that a little bit clearer. So here what we see is the divisions. So again, as I was mentioning, the sensory nuclei are located in more of the posterior compartment. So this would be the posterior horn, this would be the lateral horn, and this would be the anterior gray horn. And the gray matter, again, is just neuron cell bodies. So unmyelinated neuron cell bodies, which can communicate directly with the sensory and motor command. So if we look at the somatic sensory here, they're going to predominantly be a pain receptor, pressure from the skin, so anything sensing from the feet, stepping on a nail, stubbing your toe, things like that. The visceral is also going to be sensory. It's going to be receiving the stretch receptors and smooth muscles, such as the digestive system, the stomach wall, small intestine, large intestine. And when we come down to the anterior aspect, what we're going to see is more the autonomic motor control. So autonomic motor control are associated with the laterals, so they're closely associated with the lateral horns. They innervate the smooth muscles, the cardiac muscles, any glandular muscles. But if we stick with digestion, the sensory information from the viscera eventually cause the autonomic nervous system to start contraction of digestion in the stomach. And then we have the somatic. Somatic's pretty easy. It's what's going to control all of the contraction of skeletal muscle, so voluntary contraction. So if we just look at, a general pathway, say for instance, let's just use the somatic. Say for instance, your body senses the beginnings of the stretch receptors inside of the muscle tissue. It's going to have its own pathway. It's going to be traveling along eventually into the somatic region, so the somatic zone of the gray matter. It can also, these sensories are connected to the white matter or the track. That information is going to go up to the brain, and eventually what we're going to see is it's going to come down and it's going to target, so as it comes down from the brain, so a descending pathway, it's going to target on that particular neuron with an action potential. That action potential will then leave through the somatic neuron and eventually go into an effector. So you can think of this as like a pathway system. Say for instance, when we're talking about the visceral, so let's use the visceral pathway. Your body senses that you've eaten a large meal. Eventually it's going to hit the ascending pathway to the brain. Your brain is going to process that, and as it comes down, it will hit the autonomic region. So now as it descends, the action potential says contract the stomach, coming from the brain, and then it's going to be following a separate pathway, the autonomic motor neuron, and then eventually it's going to hit the stomach and cause contraction. So this is just a general overlay of how these tracks or pathways work and the divisions of each of the gray and the white matter. And finally what we see is just a transverse view. So here we have all of the different components of the meninges, and you can see it at roughly about this level. I'll clear this out. As soon as we see the nerve exiting, this is where we get the term spinal nerve or mixed nerve, is once it exits out, mixed nerve means that it contains both a sensory and a motor neuron, and it's going to have the different components. So again, as information is coming in through the rami, if it's a sensory, it's going to go through the posterior aspect, and then once it receives the information, say for instance from the skeletal muscular system, it's going to exit out the anterior, and it's going to still exit the same spinal nerve where the original sensory information came in. There's also accessory nerves, so we can see here, you have a posterior ramus that maybe is going into the deep muscles of the back, like the erector spinae. So you're getting sensory information about your posture, which would then eventually, as we go through the cycle again, eventually cause you to maybe exit out and have that ability to contract the erector spinae to change your postural effect. Another interesting aspect, these sympathetic trunk ganglion, they're connected. So these sympathetic trunk ganglion, if we would go up, say to the next vertebrae, there'd be another sympathetic trunk ganglia, and they're interconnected. So I'll draw one down here. So these actually are interconnected, and if we keep going down the thoracic, particular regions of the thoracic, these sympathetic trunk ganglion are interconnected. So say for instance this one is T, let's say 4, And I keep going down. This is T5 and T6. These sympathetic trunk ganglion, if the body decides to operate with the sympathetic nervous system, say for instance in a fight or flight scenario, the sensory information will still come in as usual, and then we see it, the motor response comes out, and then somewhere in the pathway it'll still have the motor command for, the effector of moving your legs, running away from something, but it can also trigger the respiratory system and increase your breathing. So it can cause all the muscles of your intercostals to start activating to increase the breath. And then the sympathetic trunk ganglion is actually correlated with or connected to heart rate control too. So that is the end of part one. Very simple. And thank you for listening, and have a good day.